This is week 14, abnormal prenatal diagnosis and termination of pregnancy, infertility, miscarriage, and art. The learning objectives for this week are found on this slide and on the next slide. And here are the final part of the learning objectives. Assisted reproductive technologies are methods that replace the source of a male or female gamete, they aid fertilization, or they provide a uterus. They've been developed so they can treat infertility, but they're also becoming part of genetic screening. The United States government does not regulate ARTs. However, the British government does. Infertility is the inability to conceive a child after a year of frequent intercourse without contraceptives. The term subfertility is used by some specialists to distinguish those individuals and couples who can conceive unaided, but from whom this may take longer than average. On a more personal level, infertility can be seemingly endless, with monthly cycles of raised hopes and crushing despair. Let's remember that as a woman ages, fertility declines. By the start of menopause, which is about the age of 50, only 1,000 or so oocytes remain in her ovaries, and spontaneous abortion occurs in 15% of women under 35, in 20 to 35% of women between the ages of 35 and 45, and up to 50% of women over 45 who try to become pregnant. The incidence of pregnancy-related problems rises with maternal age, and these, of course, include the non-disjunction, fetal deaths, premature births, and low birth weight babies. Older men are at increased risk of having children who develop autism or schizophrenia, and sperm motility declines with the age of the man. So statistically, about 1 in 6 couples has a difficulty conceiving or giving birth to children. And according to the American Society for Reproductive Medicine, males and females contribute about equally to infertility, with 25% of infertile couples having 2 factors at play. When a physical problem is not obvious, the cause is usually a mutation or chromosomal aberration that impairs fertility in the male. A common combination that accounts for low fertility is a woman with an irregular menstrual cycle and a man with a low sperm count. In looking at male infertility, we see that it's easier to detect, but often it's harder to treat than female infertility. Now, looking at how many, statistically, men are infertile, it's about 4 in 100 men. Most cases of male infertility are genetic, and some of those causes are azoospermia, low sperm count, a malfunctioning immune system, a varicose vein in the scrotum, or structural sperm defects. Now, if we take a look at the cases of infertility that are genetic, it's usually due to small deletions of the Y chromosome. And these deletions then remove genes important for spermatogenesis, or mutations in genes for androgen receptors or protein fertility hormones, or that regulate sperm development. In cases of low sperm count, sperm can be stored, frozen, then pooled together. Sperm's inability to move may be due to hormone imbalance. Abnormal shapes may reflect impaired apoptosis that removes such sperm. Genetic package of an immobile or abnormally shaped sperm can be injected into an oocyte to fertilize. The sperm cell's head is streamlined so that it facilitates movement. A misshapen sperm cannot fertilize an oocyte. It has been found that fewer than a tenth of a percent of men who are infertile have sperm cells that lack the tip called the acrosome, which contains the enzymes to break through the layers surrounding the oocyte. And in fact, an Ashkenazi Jewish family led researchers to a gene that, when mutant, causes an autosomal recessive form of male infertility due to sperm with round rather than oval heads. The family went to a center for reproductive medicine in the Netherlands, and of the six sons in the family, three were infertile. Four daughters were fertile. The affected sons' sperm were misshapen, and the mode of inheritance was recessive because the parents were both fertile. Researchers at first suspected consanguinity, but the family denied knowing a relative who had married a relative, reasoning that perhaps DNA could reveal consanguinity that the family didn't know about. Researchers scanned the genomes of all six sons for regions of homozygosity that would indicate that they had relatives marrying relatives not too far back in the family tree. A region of homozygosity in this case was defined as 25 consecutive SNPs that were homozygous. The genomes of all six sons were riddled with these regions, suggesting that at some point a cousin married a cousin, or an aunt or uncle wed a nephew or niece. One region of homozygosity was seen in all three infertile brothers, but was heterozygous in two of the three fertile brothers. The remaining brother was homozygous wild type for the region. In looking at female infertility, abnormalities in any part of the female reproductive system can cause infertility. Many women with subfertility or infertility have irregular menstrual cycles, as I stated before, and this makes it difficult to pinpoint when conception is most likely to happen. Tracking ovulation cycles aids in the determination of most likely days for conception. This slide is showing you the sites of reproductive problems in females, which I'll be discussing in a moment. You can see the pituitary tumor, the fibroid tumors. You can see the cervical cancer, anti-sperm secretions, endometriosis, scarred and blocked tube. Could happen from, say, an infection, even something like an STD. And we also have in this picture an ectopic pregnancy in the fallopian tube where the baby has started to develop. So if a woman had infertility due to the fact that she had a hormonal imbalance, then this could be caused by a tumor in the ovary or a tumor in the pituitary gland. It could be caused by an underactive thyroid gland, and it could also be caused by the use of steroid-based drugs. Fertility drugs stimulate ovulation, but they may induce the release of multiple oocytes. A woman's ovaries who are inactive or absent can use a donor oocyte for pregnancy. Some of the signs of having a reduced ovarian reserve are an ovary with too few follicles, or elevated levels of FSH, or follicle stimulating hormone, on the third day of the menstrual cycle. Blocked fallopian tubes can result in ectopic pregnancies. Excess tissue growth in uterine lining may make it inhospitable for an embryo, and this could come from such things as benign tumors called fibroids, or it could also be caused by a buildup of the uterine lining, which is endometriosis. Sometimes secretions in the vagina and the cervix may be hostile to sperm, and sperm are then not able to fertilize. And then infertility may also result if the oocyte fails to release sperm attracting chemicals. In going for a workup for infertility, the man is checked first because it's easier, less costly, and less painful to obtain sperm than oocytes. The sperm are checked for number, sperm count, motility, movement, and morphology, shape. A gynecologist can then check the female to see if reproductive organs are present and functioning. And then there may also be psychological factors that may be at play. Many people with fertility problems use alternative ways to conceive. Several of the arts were developed in non-human animals. In the United States, about 1% of the 4 million births each year are from arts. If we look at the different types of assisted reproductive technologies, we can see that Intrauterine insemination, IUI. It's the oldest art in which a doctor places donated sperm into the woman's cervix or uterus. Surrogate motherhood, in vitro fertilization, IVF, gamete intrafallopian transfer, GIFT, zygote intrafallopian transfer, ZIFT, oocyte banking and donation, pre-implantation genetic diagnoses, PGD, and sequential polar body analysis. This slide is showing some of the landmarks in reproductive technology that have occurred in the past few centuries. This is the second slide showing landmarks in reproductive technology. And this is the final slide showing landmarks in reproductive technology, which stops at 2013. Let's look a little bit more at these different types of arts. The success rate in the intrauterine insemination is about 5 to 15%. It's the first reported pregnancy from this came in 1790. And 1953, methods for freezing and storing sperm were developed. And there actually are sperm catalogs that list personal characteristics such as the color of someone's skin, their build, their educational level, and their interests and their blood type. And if a couple desires a child of one sex, such as a daughter, to avoid passing on an X-linked disease, sperm can be separated into fractions enriched for X-bearing or Y-bearing sperm. In looking at surrogate motherhood, if this is what is desired, a woman carries the pregnancy to term for another woman who cannot conceive and or carry the pregnancy. Custody rights are given up at birth, and there's complex legal and emotional issues that must be considered in this form of a birth, and a surrogate mother may or may not have contributed an oocyte. In looking at in vitro fertilization or IVF, this is where a sperm fertilizes an oocyte in a culture dish, petri dish. Embryos are transferred to the oocyte donor's uterus or a surrogate's uterus for implantation. In 1978, Louise Joy Brown was born in Britain. She was the first IVF child, and today there are over 4 million IVF children who have been born this way. A woman can undergo IVF if her ovaries and uterus are functioning, but her uterine tubes are blocked. Oocytes from the ovary are removed through laparoscopy and transferred to a petri dish. Chemicals, the sperm, and other cell types similar to those in the female reproductive tract are added to the culture. An acidic solution may be applied to the zona pellucida, in which is the layer that's around the egg, to thin it out to ease the sperm's penetration. Sperm that cannot readily enter the oocyte may be sucked up into a tiny syringe and micro-injected into the female cell. And this technique is called intracytoplasmic sperm injection, or ICSI. It is actually more effective than just doing the IVF alone and has become standard at some facilities. The ICSI is helpful for men who also have low sperm counts or have a lot of abnormal sperm. It makes fatherhood possible for men who cannot ejaculate, such as those who have suffered spinal cord injuries. ICSI has been performed on thousands of men with about a 30% success rate. In about 2 to 5 days after the sperm wash over the oocytes in the petri dish, or they're injected into them, a blastocyst is transferred to the uterus. If the hormone human chorionic gonadotropin appears in the woman's blood a few days later and its level rises, the woman is pregnant. In looking at this slide, this is showing intracytoplasmic sperm injection. And this is where a single sperm cell can be seen being injected into the cytoplasm of an oocyte. In the past, several embryos were implanted to increase the success rate of IVF. Unfortunately, this led to a lot of women having multiple births, which are riskier than single births. In some cases, the physicians actually had to remove embryos to make room for others to survive. And today, the techniques that are used in IVF have become more successful. They've improved. And currently, there are guidelines that suggest only transferring one embryo, and the rest of the embryos can then be frozen and could be utilized later. And the freezing just takes a few hours. And the oldest pregnancy using a frozen embryo occurred about 20 years after the freezing occurred. Overall, the chances of a live birth following IVF are about 20 to 35% per cycle, but the rate varies depending on certain factors that lower the likelihood of success. And these factors include maternal age. Success is 30 to 40% for women under the age of 34, but only 5 to 10% for women over 40. Um, how long a woman has actually been infertile, the number of previous failed IVF attempts, um, number of previous IVF attempts, use of a woman's own oocytes rather than the donor's, and infertility with a known cause. Several websites assess risks and predict IVF success. In one example, a couple had been infertile for 11 years. They attempted IVF 4 times that resulted in 2 failures and 2 spontaneous abortions. They had used the woman's eggs and ICSI because too many sperm were abnormal. The chance of success per IVF attempt is about 8%, but if they use a donor oocyte, it doubles. IVF may fail because of the artificial environment for fertilization. So a procedure called gamete intrafallopian transfer improves the setting. The fertilization is assisted in gift, but it occurs in the woman's body rather than in a petri dish. So what happens is that several of a woman's largest oocytes are removed. The man submits a sperm sample and the most active cells are separated from it. The collected oocytes and sperm are deposited together in the woman's fallopian tube at a site past any obstruction that might otherwise block fertilization. Gift is about 22% successful, but it also is dependent upon the woman's age. Now, a variation of gift is zygote intrafallopian transfer, or ZIFT. And what happens in this procedure is that an IVF ovum is introduced into the woman's fallopian tube, allowing the fertilized ovum to make its own way to the uterus increases the chance that it will implant. ZIFT is also 22% successful. And GIFT and ZIFT are done less frequently than IVF. These procedures may not work for women who have scarred uterine tubes. And the average cost of GIFT or ZIFT is $15,000 to $20,000. So oocytes, like sperm, can be stored frozen. Women can store their own oocytes to have children later or prior to undergoing chemotherapy. They're frozen in liquid nitrogen. And some of the difficulties exist as oocytes pause in meiosis II until fertilization occurs. And only 3% are actually successful. Now, there is an alternative to freezing oocytes. You can freeze strips of ovarian tissue that can be stored, thawed, and reimplanted at various sites, such as under the skin of the forearm or abdomen or in the pelvic cavity near the ovaries. The tissue ovulates, and the oocytes are collected and fertilized in vitro. The first child resulting from fertilization of an oocyte from a reimplanted ovarian tissue was born in 2004. The mother, age 25, had been diagnosed with advanced Hodgkin's lymphoma. The harsh chemotherapy and radiation cured her cancer but destroyed her ovaries. Before her cancer treatment, 5 strips of tissue from her left ovary were frozen. Later, several pieces of ovarian tissue were thawed and implanted in a pocket that surgeons crafted in one of her shriveled and deformed ovaries near the entrance to the fallopian tube. Menstrual cycles resumed, and shortly thereafter the woman became pregnant with her daughter, who was healthy. So freezing ovarian tissue may become routine for cancer patients of childbearing age. Women who have no oocytes or wish to avoid passing on a mutation can obtain oocytes from donors, who are typically younger women. Some women become oocyte donors when they undergo IVF and have extras. The potential father's sperm and donor's oocytes are placed in the recipient's uterus or uterine tube, or fertilization occurs in the laboratory and the blastocyst is transferred to the woman's uterus. A program in England funds IVF for women who cannot afford the procedure if they donate their so-called extras. The higher success rate of using oocytes from younger women confirms that it is the oocyte that age affects and not the uterine lining. Embryo donation is a variation on oocyte donation. A woman with malfunctioning ovaries but a healthy uterus carries an embryo that results when her partner's sperm is used in an intrauterine insemination of a woman who produces healthy oocytes. If the woman conceives, the embryo is gently flushed out of her uterus a week later and inserted through the cervix and into the uterus of the woman with malfunctioning ovaries. The child is genetically that of the man and woman who carries it for the first week, but is born from the woman who cannot produce healthy oocytes. Embryo donation includes use of the IVF so-called leftovers. In another technology, cytoplasmic donation, older women have their oocytes injected with cytoplasm from the oocytes of younger women to so-called rejuvenate their cells. Although the resulting children conceived through IVF appear to be healthy, they are being monitored for potential problem. Heteroplasmy are two sources of mitochondria that we discussed in GEN615. Researchers do not know the health consequences, if any, of having mitochondria from the donor cytoplasm plus mitochondria from the recipient's oocyte. These conceptions also have an elevated incidence of Turner syndrome, 45XO, which often causes spontaneous abortion. Because oocytes are harder to obtain than sperm, oocyte donation technology has lagged behind that of the sperm banks. One IVF facility that has run a donor oocyte program since 1988 has a brochure that describes 120 oocyte donors of various ethnic groups, like a catalog of sperm donors. Pre-implantation genetic diagnosis, PGD, is often teamed with IVF to detect genetic and chromosomal abnormalities before pregnancy starts. The procedure is called diagnosis if it detects a specific gene variant known to be in one or more of the parents, and screening if it is used to construct a karyotype without looking for a specific chromosomal anomaly. The couple selects a very early pre-implantation embryo that tests show has not inherited a specific detectable genetic condition. Pre-implantation refers to the fact that the embryo is tested at a stage prior to when it would naturally implant in the uterus. PGD has about a 29% success rate. One cell or blastomere of an 8-cell embryo can be removed for testing. The slide is showing pre-implantation genetic diagnosis, PGD. If you are to look at A here, on this picture, it's a single cell is separated and tested to see if it contains a disease-causing genotype or atypical chromosome. And in B, if it doesn't, the remaining 7 cells divide a few more times and are transferred to a woman's uterus, typically the oocyte donor, to complete the development. And here in C, you see the removal of the cells. Now, PGD is not new. The first children were born following using PGD in 1989. In these first cases, probes for Y chromosome specific DNA sequences were used to select females who could not have inherited the X-linked conditions their mothers carried. The alternative to PGD would have been to face the 25% chance of conceiving an affected male. In March 1992, the first child was born who underwent PGD to avoid a specific inherited disease. She was checked at an 8-celled pre-implantation embryo to see if she had escaped the cystic fibrosis that affected her brother. Since then, PGD has helped to select thousands of children free of many types of single gene conditions. Like most arts, use of PGD has expanded as it's become more accurate, and it has also become more familiar. This slide is summarizing the different procedures and with the different technologies that are available at this time. So looking at sequential polar body analysis, this technique may substitute for PGD, and it provides genetic information even earlier in development. The approach is based on the fact that meiosis completes in the female only as a secondary oocyte is fertilized. So if a woman is heterozygous for mutation, then an oocyte will contain the mutation and its associated polar bodies will have the wild type allele or vice versa. This is in accordance with Mendel's first law of gene segregation. The timetable of female meiosis is important too. The first polar body forms as a developing oocyte leaves the ovary. That polar body is not accessible and it will not show the effects of crossing over. A second polar body, however, which forms at fertilization, can be tested. Researchers can sequence link markers or even entire genomes of second polar bodies to determine whether crossing over has occurred, to be certain that the fertilized ovum has not inherited the mutation. Because the sequential polar body analysis is still experimental, researchers follow up with PGD to test their predictions and to ensure that the IVF embryos transferred to a woman's uterus to develop are free of the family's mutation. The idea of probing polar bodies dates from the 1980s, but the techniques first led to results in 2011. So far, mutations behind more than 150 single gene diseases have been detected at this initial stage of prenatal development, and that's just the very first cell. The slide here is showing you the sequential polar body analysis, the technique here with the fertilized ovum. You can see the pronuclei and you can see the second polar body. So people want to know the fates of the frozen embryos. And when you look at these extra oocytes, fertilized ova, or very early embryos, they can be stored indefinitely. They can be stored and destroyed after a set time. They can donate for embryonic stem cell derivation and research. They can be thawed later for use by the biological parents, or they can be thawed later for use by other parents. Or lastly, they can be discarded. In the United States, nearly 500,000 embryos derived from IVF sit in freezers. Some have been there for years. Most couples who donate their frozen embryos to others do so anonymously with no intention of learning how their genetic offspring are raised. However, some couples keep track of the embryos, helped by social media to make connections with potential other parents. One couple who had twins from 2 of 18 IVF embryos chatted online with and selected another couple to receive the remaining embryos. That couple also had twins, and they then shipped the remaining frozen embryos to a third couple who also had twins. Extra frozen embryos are also an excellent resource for same-sex couples to have children with the help of a surrogate mother if necessary. Another alternative to disposing of fertilized ova and embryos is to donate them for research. and the results of experiments sometimes challenge long-held ideas, indicating that we still have much to learn about early human prenatal development. And this slide here is showing how some of those were transferred between the three families here. It is important, though, to realize that arts introduce ownership and parentage issues, and the human genome information is providing more traits to track and perhaps control in coming generations. So, who will decide which traits are worth living with and which ones are not? So, on this slide here, you can see some of the assisted reproductive disasters. And I just want to go down to number 3 here because several couples planning to marry discovered that they were half siblings. Their mothers had been inseminated with sperm from the same donor. Well, way back and even in the 1970s when I was in graduate school, there were men who were going to Georgetown Hospital and they were selling their sperm, just like people used to sell blood. So they were selling their sperm, and even though they called them sperm donors, they were in actuality sperm vendors. And so that this was a practice that was done. And the other thing is that these were sperm from graduate students, quite often in biology, and or they were medical students. And then they weren't shipping the sperm out of the area; they were keeping it within the specific area. So in that case, it was Washington, D.C., but it was also when I moved to work in Philadelphia, I saw that the same thing was going on there, where they were keeping it in the Philadelphia area, so that you could see that this could be a possibility in the future. The other issue is that they weren't asking questions to these individuals who were selling their sperm, like, what is your heritage? You didn't do a family history on them. They didn't ask them about specific diseases, and they weren't able at that time to screen them. They did not, I shouldn't say weren't able, they did not screen people for specific diseases by doing carrier testing, such as Tay-Sachs and sickle cell, for just an example of two of them. And here are three more cases that you might find interesting to look at. I thought that was quite interesting. If I look at number 6, man sues his ex-wife possession of their frozen fertilized ova. He won, and then he donates it to research. She had wanted to be pregnant. So, I mean, some of these things are just like soap operas, but I just included them in here.